welcome to this lesson for Advanced Higher Biology. We are in Unit 2, Key Area 1, and we are looking at a sampling of wild organisms. So previously we've just had a wee look at how to be safe when you're carrying out fieldwork, and today we're going to focus specifically on what you might be doing when you're out in the field. We will then move on to look at how you may use what you've collected and how you might identify those particular organisms. So for today, we would need you to understand how you can correctly sample wild organisms in their environment. So you need to be able to explain why sampling is necessary, describe why and how to minimise the impact of sampling, list and describe the use of different sampling techniques, and explain the use of specific techniques for specific organisms. So why do we need to sample? Now, those of you who have already looked at Unit 3 may be a little bit more familiar with sampling than others. This does come up when we're looking at designing an experiment. But I'm going to assume for the purposes of the video that you haven't seen that section yet, or you might not remember it. So really we consider sampling because the most thorough biological studies would really be ideally able to gather data from all of the individuals within a population. Now that is often quite impractical due to the time and the cost considerations. So sampling can eliminate the need to measure and record every member of a population as long as the sample is what we consider to be representative. So it's important to have, if you have, for example, males and females in a population in a particular ratio, your sample should focus or have a similar ratio to your original sample population. So that's really what we're looking at when we consider a sample population being representative. It should feature the same amount of variation as your original population. Now, it's obviously very important to consider whether the sample that is being measured, so the organisms that you're looking at or the area that you're in, if that's going to be affected by the actual sampling procedure itself. And if you are impacting on your organisms, is that going to affect your results and conclusions? So when you're designing your study, you need to take these into consideration. Now, if you are carrying out a study that involves more invasive techniques, then that particular study really should have benefits that are going to outweigh any particular negative impact that might be seen on the organism. We also need to think about whether or not sampling will involve any rare species or habitats which are considered to be vulnerable and are therefore protected by legislation. Sampling in either of these cases could potentially be illegal unless it's carried out under specific licenses that might be awarded to scientists when they're going in to look at things. So a rare species would be one which is found at a lower density or the total population is small. A vulnerable habitat would be one which is easily damaged by human activity. And in Scotland we have several protected species like the red squirrels, deer, red kites, basking sharks, and many of our wild plant species are considered to be protected. Now, when it comes to our sampling methods, there are several different ones that could be selected. And using the correct one really depends on the type of organism that you are sampling. So we're going to focus today on sampling methods for birds, plants and small mammals. We're going to focus around those, generally speaking. To begin with, we're going to have a wee look at what is called a point count. This is often used for bird species. So a point count is one which is carried out from a selected stationary location, and we record all of the individual organisms which are observed. So it's used for determining any species abundance and would be used to potentially the data could be compared to other point count locations or with data from the same location gathered at a different time. So you may be standing in that particular area counting how many birds, individual birds of a particular species that you see one day and then coming back and doing it another day, or you may be sampling two different areas on the same day, for example. Now, counting birds in particular can be quite frustrating. Often birds show territorial behaviour and that keeps populations quite usually quite uniformly distributed, but also quite thinly distributed. You don't find too many members of the same species near each other, and consequently it can be quite difficult to see them when you're carrying out a point count. Obviously, you're also in a more dense vegetative area, so it can be quite difficult. Visibility can be a little bit poor. So you might have one observer counting all the birds seen or heard during a particular time within the certain area. And it might be quite important to know whether or not this has been done either on what we would refer to as a limited radius. So have they mapped out a specific area, and if they see a member of the species beyond that area they wouldn't count them, or are we using an unlimited radius whereby they're just looking to see count as many individuals of the species as they can possibly see. So that's relatively important and gives us an idea of rough idea even of how many of a population we have of that particular species. Now, quadrats and transects are our next particular technique, and you'll be fairly familiar with both of these. These are used for plants or either sessile or slow-moving organisms, and a quadrat and a transect are often used together. So often if you're working on a transect line, you'll be using a quadrat to count your individual organisms. So a transect is a line along which our different samples can be taken. They're often set up along an area where the terrain or the abiotic factors are changeable. So for example, a woodland that's running into a field, if we're working up the way up sandy shore from the water line to the high tide line, for example. And we're going to be looking at abiotic factors like moisture, light, pH, salinity, those sorts of factors. So when you have your transect line laid out, you're going to take your quadrats of a suitable size and shape, place them along the transect and allow the abundance of whichever organism it is you are looking at to be recorded. You're also going to be using any kind of meter, whatever it is you're measuring, to measure your appropriate or relevant abiotic factor, and comparing the two pieces of data together will give you an idea of how your organism responds to the abiotic factor that you're measuring. Now you will of course be very familiar with the quadrat, and it ensures that our standard area is being sampled each time a measurement is made. And obviously having a standard area helps to ensure that we're using the same amount of effort at a sample point and helping to reduce our bias and increase reliability. Now you might have slightly different sizes of quadrat, and that's going to be determined, the use of which one will be determined by how uniformly your organisms are distributed in your habitat. A smaller quadrat is going to be obviously more suitable where an organism is small or very densely packed together, and the diversity of your environment is going to influence how many samples specifically that you need to take. When it comes to mobile species, we really need to think about capture techniques such as nets or traps. So birds or bats could potentially be caught in mist nets, although again that quite often needs to be done under license as it could have an impact on the organism. Night flying insects might be sampled using a light trap. And a small mammal could be potentially caught in a longworth trap and caught and released. So again, when setting these up, you need to be very careful that you are not causing any adverse harm or impact to the organism. Some animals are going to be very difficult to sample through these normal observation techniques, so we would consider these to be fairly elusive species, and there's a few methods we can use in order to record their presence or absence. We could use camera trapping to directly sample these elusive species. A camera trap is a remotely activated camera equipped with a motion sensor or an infrared sensor, or potentially could use a light beam as a trigger. And so we would often look at research applications including detection of a rare species, estimating the population size and the species richness. The issues with cameras are that they could potentially be damaged due to the weather or the animals corrupting the footage. We have also got to use sometimes indirect methods of sampling, so we would consider that as scat sampling. So that would be looking at the animal's droppings, and that's going to be, they're going to be collected in a particular area and will often give us an idea of the information about the species abundance, and also would give us some information about the species diet. So if you can't reliably use a camera trap for whatever reason, this would count as evidence of the presence of the organism that you're looking for. The disadvantages are in terms of identification, in terms of making sure you've seen them, and you're also assuming that the population of a species, not all animals of the same species are necessarily going to have the same diet, so you may not actually identify appropriate droppings where you should be. You might be missing some or counting some which shouldn't be counted, so it can be a bit less reliable. So from that, you should understand the various techniques we may use to sample wild organisms in their environment, explain why it's necessary, why and how we need to minimise the impact of our sampling, the different sampling techniques that you might use, and why you might use different techniques for different organisms. As ever, please get in touch if you have any questions, either by leaving a comment, visiting the website, or getting in touch via the email address to follow.